Hi, and welcome to the iBio lecture called Chemical Glycobiology. My name is Carolyn Bertozzi, and I'm a professor of chemistry and molecular and cell biology at the University of California at Berkeley, and also an investigator of the Howard Hughes Medical Institute. I am a chemist by training who became interested in the biology of sugars, which is what the term glycobiology means, when I was in graduate school and then later as a postdoctoral fellow. And my laboratory research at UC Berkeley seeks to combine chemistry and biology together to understand what sugars are doing in the human body. So let me begin by giving you an introduction as to why I'm so interested in the biology of sugars, along with all of the students and postdoctoral fellows that work with me in the laboratory. It turns out that around the turn of the millennium, there was a very exciting sequence of breakthroughs in biology having to do with sequencing genomes. So in the early days of genome sequencing, the first eukaryotic organism to be characterized in this way was the budding yeast. And one of the great surprises from the sequence of the yeast genome was the fact that it only contains about 6,000 genes. And that's not a very large number. In fact, before the genome was completed, there were some estimates that there would be far more genes required to encode all of the interesting functions that eukaryotes perform. So the number 6,000 seemed very small. Of course, the budding yeast is a relatively simple eukaryotic organism. It's a single-celled organism. And there was this idea that more cells would require more genes. And that seemed to be the case as more and more genomes were decoded. So when the C. elegans genome was finally sequenced, that genome had about 15,000 genes, which is a larger number of genes, and it seemed to make sense. And then the next major organism to have its genome sequenced was Drosophila, or the simple fruit fly, which had about 20,000 genes. And all the while, scientists were working on the human genome, operating under the assumption that the human genome would be much larger than any of these other model organisms. In fact, some early scientists estimated that the human genome might have over 100,000 genes. Well, around the turn of the millennium, there was a big surprise yet again in the world of genome sequencing when the human genome was finally completed, and it turns out that it's not that much larger than the fly genome or the C. elegans genome, and really not even that much larger than the yeast genome. There are only around 25,000 genes encoded in the human genome. These are the genes that encode for proteins. And by the way, I think that nobody would be more surprised than Charles Darwin himself to discover that we really don't have that many genes. So then the next question became, given that we only have about 25,000 genes, how is it that human beings can be so biologically complicated compared to some of these other simple model organisms? Well, in one of the major publications in 2001 that put forth the sequence of the human genome, Craig Venter and his colleagues made this statement, which is that the finding that the human genome contains fewer genes than previously predicted might be compensated for by combinatorial diversity generated at the level of post-translational modification of proteins. So, put another way, what happens in the higher, more complicated organisms is that the proteins encoded by the genome are modified in very complicated and diverse ways to create a lot more biological complexity than one might predict just by simply counting the genes in the genome. And modifications of those proteins are what we call the post-translational modifications. Well, it turns out that one of the most complicated of those post-translational modifications is the attachment of sugars to those proteins. And that's the process that we call glycosylation. And by the way, you'll see that prefix glyco again and again and again throughout these lectures. That's the Greek prefix for sugar. It turns out that many of the proteins that are glycosylated are proteins attached to the membranes of cells. In fact, they reside on the surface of cells, and we call those sugar-modified proteins glycoproteins. And an example of one of those glycoproteins is shown here in cartoon form, where the protein is this blue structure that's anchored in the membrane of the cell. We also have lipids in our cell membranes that have sugars attached to them. So for example, this cartoon illustrates what we call a glycolipid. And the sugar part of the glycoprotein or the glycolipid is what we might refer to scientifically as a glycan. Glycan is just another word for a complex sugar molecule. So because of all of these glycoproteins and glycolipids on the surface of our cells, we basically can think of our cells as having a sugar coating. And in fact, that's why on the very first slide of this lecture, I had a cartoon of M&Ms. Because in many ways, you can think of our cells as being like M&Ms. They're coated with sugar molecules. Now, that to me is fascinating. And of course, it begs the question, what are the functions of all of these sugar molecules that are attached to the proteins and the lipids and decorating the surfaces of our cells? And that question, what are the functions of the sugars? That is the question that's embodied by the field of glycobiology. Now, it turns out that unlike M&Ms, which have a fixed sugar coating that never changes, of course, until you eat the M&M, the sugar coating on the surface of our cells does change. And it can change dramatically as our cells change their state. Now, we have terms that we use to describe the collections of these sugars. In fact, we have this term glycome. You can think of this term glycome as being analogous to other terms that you might be more familiar with. Genome, for example. The genome is the complete collection of all of the genes in our cells. And maybe you've heard the term proteome. The proteome is the complete collection of the proteins that our cells are making. Well, in the field of glycobiology, we use the term glycome to describe the totality of glycans produced by a cell. And as I'm showing here in this cartoon, the glycome is dynamic. So in other words, when a cell has one particular state, it'll have a particular collection of glycans, which are shown by these various cartoon structures. But if the cell undergoes a physiological change, the collection of glycans can change. Some of the structures might become more abundant or less abundant, and there might be entirely new glycan structures that weren't present in the original state. So it's the dynamic nature of the glycome that is so interesting from the perspective of understanding what these sugars are doing in biology. Just to give you some examples of situations in which the glycome changes, if you look at the complete collection of glycans when a cell is in an embryonic state, so it's a cell that has just formed, you'll see a certain collection that's quite different from the collection of glycans when that cell is in what we call a differentiated state. In other words, when the cell has chosen to become a muscle cell, or a neuron, or a skin cell. Each of those cell types has its own distinct glycome, which is different from the embryonic cell that it came from. Turns out that the glycome also changes during diseases. So if you look at the complete collection of glycans when a cell is in a healthy, normal state, it's different from the collection of glycans when that cell becomes a cancer cell. Now, this is a very interesting discovery from the perspective of clinical medicine, because if we could actually see how the glycome changes on cells in the human body, we might be able to detect cancers. And for that reason, as you'll see later in my second lecture, we're very interested in developing tools to image the glycome, to see the glycome inside a living body. So keep that in mind. Now, let me tell you a little bit about where these glycans come from inside the cell, because they are the products of fairly complicated metabolic pathways. They're products of metabolism. And all of that begins with the uptake of simple sugars into the cells. And these simple sugars we call monosaccharides, and they're denoted by these little colored balls. These are simple sugar molecules. So you eat food, the food has sugars in it, and your cells take up those simple sugars. Now inside the cell, the monosaccharide building blocks are processed by enzymes. Eventually, those building blocks are sent into subcellular compartments that we call the endoplasmic reticulum, or the ER, and the Golgi compartment. And these membrane-bound organelles are basically an assembly line for the construction of complex glycans from simple monosaccharide building blocks. So the glycans are built inside the ER and the Golgi, attached to either proteins or lipids, and then eventually those proteins and lipids, or glycoproteins and glycolipids, are delivered to the plasma membrane where the cells are now coated with these sugar molecules. Let me tell you about the monosaccharide building blocks. And first of all, I should say that there are many of these sugars in nature, and different organisms have different collections. So I'm only showing you the monosaccharides that you would find in vertebrate glycans, which are distinct from the monosaccharides you would find in bacteria or even plants. But these are the ones that we have inside our bodies. And there are 9 of them. So this is a good number to know. There are 9 monosaccharide building blocks, just like there are 4 nucleotides in your DNA or 20 amino acids in your proteins. And each of these monosaccharide building blocks goes by a different name, and we also have abbreviations that we use to denote them very quickly. So for example, many of you are familiar with this sugar called glucose. Glucose is really the parent of all of the other monosaccharide units. In fact, your cells can build any of these other building blocks starting from glucose, if it had to. And glucose goes by the abbreviation GLC, and we often just say glyc, a simple little word to denote glucose. And then some of these sugars are perhaps more exotic in their structures. For example, this one. This monosaccharide is called sialic acid. It has more carbon atoms than the other sugars. It also has this carboxylate. It carries a negative charge. And I'm going to come back to sialic acid later on because it occurs in some interesting biological circumstances. Now, we have terminology that we use to describe the structures of higher order glycans. These are structures that are made of multiple monosaccharide building blocks. So, for example, glucose is simply a monosaccharide, and we often think of this as a metabolic sugar. But if you take glucose and link another sugar to it, and this one is galactose, these two together make a disaccharide that's known as lactose, which you might have heard of also because it's abundant in milk. It's a milk disaccharide. It's disaccharide because it has 2 monosaccharide units. And here's a structure of what we would call either an oligosaccharide or a polysaccharide, which are terms that we use interchangeably. This is a much larger structure which has many copies of glucose all linked together in a long polymer. This structure is cellulose. It's the major component of plant cell walls. And in fact, it's the most abundant organic material on Earth. It's very important to understand the structure of cellulose. Now, when we link monosaccharides together to make these larger glycans, we need terminology to describe the nature of those linkages. In this way, the glycans are more difficult and more structurally complicated than other biopolymers like DNA or RNA or proteins. The difference is that those other biopolymers are linear. And all of the linkages, whether they're amide bonds in the proteins, phosphodiesters in the nucleotides, all those linkages are the same. But for glycans, each linkage can be different, and rather than simply being linear, the glycans can also be branched. And we also have issues of what we call stereochemistry, which has to do with the orientation of linkages. So it's more complicated. So just to give you a sense of how complicated it can be, what I'm showing here is the structure of a trisaccharide. So there are only 3 monosaccharide building blocks linked together. It's a fairly simple structure compared to some other glycans in nature. But even with this trisaccharide, we have to describe not only the orientation of how each of these sugars is linked to the next one, but also the position on each sugar to which a sugar is linked. Because as you'll see, each of these sugars has multiple hydroxy groups. And each hydroxy group could potentially be a site of linkage to another sugar. So we need to understand for each sugar both the regiochemistry of its linkage, which is the orientation, or I should say the position, as well as the stereochemistry, which is the orientation. So for example, over at the end here, there's a galactose, and this galactose is linked to the hydroxy group at the 4 position of N-acetyl glucosamine. That 1,4 linkage is the regiochemistry. And this orientation of a bond is what we call the stereochemistry, and we define this as beta. By contrast, this fucose residue is linked through what we call an alpha linkage to the 3-hydroxy group of N-acetylglucosamine. So if we take all of that information together, we would then describe the trisaccharide as galactose, beta 1,4 to N-acetylglucosamine, with at the same time, in parentheses, a fucose linked alpha 1,3 to the same N-acetylglucosamine. So as you can see, it gets pretty difficult. But there are some simple elements of the structure that are easy to remember. So with DNA, we think of a 5' end and a 3' end. With proteins, we think of an N-terminus and a C-terminus. Well, with glycans, there are also 2 distinct ends. We call them the non-reducing terminus and the reducing terminus. So at the very least, you can think of a glycan as having 2 ends. And then if you need more details about the structure, you have to understand what we call regiochemistry and stereochemistry. Okay. Now, as I said, this is a simple structure. In nature, these structures can be far more complicated. And I've taken it up a notch in this slide just to show you examples of actual glycan structures that have been found on human glycoproteins. And these are examples of 2 varieties. One we call an N-glycan. We call this an N-glycan because it's attached to a nitrogen atom on the side chain of an asparagine residue within the protein scaffold. This variety is called an O-glycan. It's an O-glycan because it's linked to the oxygen atom on the side chain of either serine or threonine that's within the protein, that's the scaffold. And as you can see, this particular N-glycan is branched. It has these 2 arms. We call these antennae. It turns out these N-glycans can have 3 antennae or 4 antennae. They can be much more complicated than this. And here, this is an O-glycan that also has a branch point and then another branch point. It's a pretty complicated structure. But one thing we've learned by looking at all of the different glycans in the glycome is that those structures are not random. In fact, elements of those structures are highly conserved in particular organisms. So in vertebrates, for example, the N-glycans can be very diverse in the parts that are out here, in the structures of the antenna. However, this part here that's close to the protein scaffold is generally highly conserved and similar from glycan to glycan to glycan. Likewise, in the O-glycan family, there's a lot of diversity out here, but this sugar is always conserved. It's always the same sugar that's linked in the same way to the protein backbone. So there are conserved and variable parts of these glycans. All right. Now, I mentioned that the glycans are assembled inside the Golgi and the endoplasmic reticulum. And there are enzymes that reside in those compartments that do this enzymatic chemistry. We call those enzymes glycosyltransferases. Now, I thought I would mention a point of historical interest, which is that the discovery of this mechanism of biosynthesis is largely attributed to Louis Leloir, who back in the 1950s discovered that glycogen, which is a storage form of glucose in vertebrate systems, is built biosynthetically from a precursor in which the glucose is linked to a nucleotide diphosphate. And we call this nucleotide sugar UDP glucose. Here's the UDP part, uridine diphosphate, and there's the glucose. Now, this was an important discovery because it suggested a mechanism by which glycans in general might be synthesized. And in fact, the importance of Leloir's discovery was recognized with a Nobel Prize. In the forward sense, the way that glycogen is assembled is through the action of an enzyme that one would classify as a glycosyltransferase. It transfers a glucose onto the growing polysaccharide. And the substrate it uses is, again, the UDP glucose. Now, it turns out that all of the glycosyltransferases, or I shouldn't say all, but most of the glycosyltransferases, use substrates that are similar to this nucleoside diphospho sugar. And I'll just show you examples from, again, vertebrate biology. So many of the sugars can be found in this UDP form, not just glucose, but also galactose, N-acetylgalactosamine, N-acetylglucosamine. whereas some of the sugars are found linked in the form of a GDP nucleoside. For example, GDP mannose and GDP fucose. And these are the substrates for their respective glycosyltransferases. And then sialic acid kind of stands alone in vertebrate biology in that its activated form is to a cytidine monophosphate, or CMP sialic acid. And there are a family of sialyltransferases that all use this as what we call a glycosyl donor. So these are the substrates that are made inside your cells and used by your enzymes. Just to give you a sense of how enzymes might assemble a tetrasaccharide, this is a pathway that is found in vertebrate systems. So this disaccharide is synthesized, and then a sialyltransferase will take the sialic acid from CMP sialic acid and transfer it onto this sugar, converting the disaccharide to a trisaccharide. Then along comes a fucosyltransferase that will transfer fucose from GDP fucose and convert the trisaccharide to a tetrasaccharide. This particular tetrasaccharide has some very interesting biological properties that I'll be coming back to later in this lecture. In the history of glycobiology, probably one of the most important discoveries that really started to attract a lot of interest from outside the field was the discovery in the middle of the last century of the human blood groups. Now, this is a discovery that has had huge implications with respect to understanding immunology, the human immune system, and also it's a discovery that was central for the development of blood transfusions. Of course, the blood transfusion is one of the most important clinical procedures. It turns out that your blood type is determined by sugars. So hopefully all of you know your blood type. I can tell you mine is O positive. Some of you might be blood type A, some will be blood type B, and some of you might be blood type AB. Well, what it means to be O or A or B or AB is simply what are the structures of the sugars on your blood cells. So for example, as someone who is blood type O, what that means is that my blood cells have this trisaccharide structure on the surface, on the glycoproteins and some of the glycolipids. That defines me as blood type O. Now, some of you are blood type A. What that means is that you also have this sugar biosynthesized in your cells, but you have an enzyme that I don't have. That enzyme transfers this new sugar onto the trisaccharide to build a tetrasaccharide. And if you have this particular tetrasaccharide on your blood cells, you're blood type A, by definition. Now, those of you who are blood type B have a slightly different enzyme. Instead of transferring this red sugar, which is N-acetylgalactosamine, your enzyme transfers the green sugar, which is galactose. So when galactose is added to this trisaccharide, you get a tetrasaccharide, which is slightly different, and this is the B tetrasaccharide. So those people are blood type B. For those of you who are really into chemical detail, if you look closely at the structure of A and the structure of B, you'll notice there's a single chemical functional group that is different between these two structures. It's very subtle. So right here in blood type A, there's an N-acetamido group, an N-acetyl group. Over here in blood type B, it's a hydroxy group. That's the only difference. And yet the human immune system is so exquisitely sensitive to structural differences that your immune system can detect the difference between these two instantly. And that's why if you have blood type A and by accident you receive a blood donation from a blood type B donor, your immune system will react against this and reject the blood, and that's a disaster. So understanding the structures of the human blood types and what that means to the immune system was absolutely critical for blood transfusions to occur. And by the way, those of you who are the AB blood type, what you have is this enzyme and this enzyme. You got one enzyme from your mother, the other enzyme from your father, and you can make a 50-50 mixture of these two structures. That's what you've got on your blood cells.